Assalamu alaikum. Previously, we have discussed that if we purify a protein from body and we want to study that protein, then the first thing we determine the composition of amino acid in that protein. The very next step is to determine the sequence of amino acids in that polypeptide chain. So, a scientist, Pierre Edman, introduced a technique known as Edman degradation technique. With the help of this technique, we determine the sequence of amino acid in that polypeptide chain. Okay, so according to this technique, a reagent known as phenyl isothiocyanate will react with the N terminal of the polypeptide chain. Okay, so we can separate the amino acid which is present at the N terminal of the polypeptide chain, and we will label that protein by the help of chromatography or electrophoresis technique. Now we will discuss the detail of Edman degradation process. Okay, now let's start. Once we know the composition of amino acid in a protein, the very next step is to determine the sequence of amino acid in that polypeptide chain. Okay, so we can determine the sequence of amino acid in a particular protein by Edman degradation process. Okay, according to this process, a reagent known as phenyl isothiocyanate. This is phenyl isothiocyanate. This has one particular characteristics that this reagent will interact with the N terminal of the polypeptide chain. Okay, so suppose we have a polypeptide chain of 3 amino acids, which is alanine, glycine and serine. Okay, now we will expose this polypeptide chain to the phenyl isothiocyanate. Okay, so this reagent will interact with the N terminal of the polypeptide chain. Okay, this will interact with the N terminal of the polypeptide chain. Okay, this will done in slightly basic condition, in slightly basic solution. Okay, now, this reagent phenyl isothiocyanate will interact this N terminal of the polypeptide chain in slightly basic solution. Okay, now the next step is to break the peptide bond between alanine and glycine. Now we will break the peptide bond between the alanine and glycine. This is done in slightly acidic solution. Okay, in slightly acidic solution. Okay, now the peptide bond between the alanine and glycine will break from here. Okay, now this label amino acid containing a phenyl isothiocyanate reagent will be separated by a technique known as chromatography or electrophoresis technique. Okay, now we will again interact this phenyl isothiocyanate with the remaining polypeptide chain to determine the next amino acid which is present in that polypeptide chain. Okay, now this process repeat again and again, and we will label the amino acid, and we label all the amino acid which is present in that polypeptide chain. Okay, so hopefully today you understand my lecture. Allah Hafiz.